Hello there, I'm Flavio Tomasi, one of the 14 early-stage researchers of the European-funded consortium SimpleSci. In this consortium, we studied the role of synaptic dysfunctions in neuropsychiatric disorders, using different approaches. I work at the Sorbonne University in Paris, under the supervision of Catalina Betancourt. In our lab, we studied the neurobiological basis of neurodevelopmental disorders, in particular autism.
Neurodevelopmental disorders are disabilities resulting from abnormal brain function. Children with neurodevelopmental disorders can have deficits in language and speech, learning, memory, social behavior, motor skills, and other neurological functions. Examples of neurodevelopmental disorders include autism, intellectual disability, and epilepsy. These conditions are common in the general population, and except for epilepsy, there are usually no pharmacological treatments. Therefore, more studies are needed to develop effective treatments. In my group, we study the potential of the endocannabinoid system as a possible target for the development of novel therapeutic approaches. The endocannabinoid system is a communication system between cells in the brain and the body that affects many important functions, including how a person feels, moves, and reacts. It was named after the plant Cannabis sativa and its active ingredient THC because it was discovered studying the effects of this molecule in 1980s. The endocannabinoid system is involved in almost all body processes and has a crucial role in maintaining homeostasis or balance within the body. This system can be seen as a control panel that reacts to internal and external stimuli to regulate general health. The molecules produced by the body that interact with the endocannabinoid system are called endocannabinoids, and they interact with two receptors, CB1 and CB2, which are present throughout the body. Also, THC from the cannabis plant interacts with these receptors. To understand the importance of the endocannabinoid system for neurodevelopmental disorders, we have to take a look at how the brain cells, or neurons, work. Neurons communicate with each other by sending chemical messages, called neurotransmitters, through the synapse, the space between two neurons. Neurotransmitters released from a presynaptic neuron travel across the synapse and attach to specific receptors located on the postsynaptic neuron. This activates the postsynaptic neuron and triggers a set of events that allows the message to be passed along. However, the endocannabinoid system communicates its messages in a different way because it works backwards. When the postsynaptic neuron is activated, endocannabinoids are released and travel backwards to the presynaptic neuron, where they attach to the endocannabinoid receptors CB1 and CB2. So why is this important? Since endocannabinoids act on presynaptic neurons, they can control what happens next time the neurons will be activated. In other words, this process controls how messages are sent, received, and processed by neurons. It is possible to interfere with this control by the endocannabinoid system with the use of pharmacological compounds that block the effects of the natural release of endocannabinoids in synapses. In my project, I work on a neurodevelopmental disorder called Phelan-McDermid syndrome. This disorder is caused by a genetic defect, the loss of a gene called SHANK3. The loss of one of the two copies of SHANK3 reduces the efficacy of the protein and alters the communication between neurons. The main manifestations of Phelan-McDermid syndrome are intellectual disability, autism, speech deficits, weak muscle tone, and impaired motor skills. In our lab, we are working with a mouse model of Phelan-McDermid syndrome. This means that we have a mouse that is genetically modified to delete SHANK3. This loss causes alterations in the behavior of these mice, but also in the molecular composition, shape, and function of synapses in the brains of these animals. The goal of my project is to check if modulators of the endocannabinoid system can have beneficial effects in this mouse model. Specifically, I am investigating the effects of novel pharmacological compounds that block the cannabinoid receptor CB1. After repeated administration of these drugs to SHANK3 deficient mice, I am studying whether the treatment is able to rescue the deficits caused by the loss of SHANK3. Specifically, I am testing the effects on behaviors such as locomotion, learning and memory, and anxiety. I am also studying the molecular composition of the synapses and analyzing the effect of the drugs on the morphology of the synapses and their function. If these drugs prove to be effective in our mice, the ultimate goal would be to test them in patients with Phelan-McDermid syndrome and hopefully contribute to ameliorate the lives of people with this disorder.
I hope you enjoyed this video, and if you'd like to discover more, please check our website and social media, and don't miss the next episodes of SimpleSci.